All right, so here we are, carbohydrate metabolism. We've already talked about glucose, which gets metabolized through glycolysis, or it, is built up with glucogenesis. With this, we're going to be talking about specifically the breakdown of glycogen, your storage molecule, as well as some other sugars. So glucose is our primary dietary sugar. Several other sugars are very important, however, to vertebrates. For instance, fructose, galactose, and mannose. Now, besides glucose, these are the most common sugars that you're going to found in oligo and polysaccharides. These are great energy sources. So the conversion of our other sugars into glycolytic intermediates is ultimately our goal. Now, if you look here, what I'd like to draw your attention to is our glucose, which is right here. So glucose, we know that we phosphorylate it, we make glucose 6-phosphate, and ultimately make our way down to pyruvate down here. So basically glycolysis is, I'm drawing my arrow all the way down there. Some other sugars can ultimately get there, for instance, galactose, fructose, mannose, and fructose over here. They can get to glycolysis, but it might take a couple of different steps. For instance, galactose gets phosphorylated to make galactose 1-phosphate. That galactose then has to be converted ultimately to make its way to glucose 1-phosphate and then to glucose 6-phosphate. And obviously there's a little bit involved to get there, but ultimately it can. Fructose, well, fructose actually just kind of jumps in to glycolysis because it gets phosphorylated by hexokinase as well. Because if hexokinase, that prefix hexo, that simply means 6. Hexokinase and glucokinase are enzymes that both phosphorylate glucose, but hexokinase also phosphorylates a number of other sugars. So fructose can get phosphorylated to make fructose 6-phosphate. Likewise, mannose can get phosphorylated by hexokinase to make mannose 6-phosphate. Then we undergo a little bit of a conversion, and fructose and mannose can kind of jump right in. So I guess kind of our goal is to get a sugar to something related to glycolysis. Now, great sources of fructose are listed here. We've got oranges, which have 9.2 grams per 100 grams of an orange. Honey is 82 grams of fructose, or sorry, total sugar. 100 grams of an orange, 9.2 grams of it is sugar. 4.6% or 50% of that is fructose. So we've got a pretty good range of different fruits, as well as maple syrup, agave syrup, for fructose. Now, how is fructose metabolized? Well, one way in which fructose is metabolized, if we look down at the bottom here, it gets phosphorylated. We make fructose 1-phosphate. Fructose 1-phosphate aldolase breaks that into dihydroxyacetone phosphate and glyceraldehyde. Now that glyceraldehyde, what's important to recognize is that glyceraldehyde is not glyceraldehyde 3-phosphate, so we have to employ another enzyme, glyceraldehyde kinase, to phosphorylate that glyceraldehyde to make glyceraldehyde 3-phosphate. So this is going to be metabolized much more quickly than than glucose. Now this can enter our glycolytic pathway in two routes, or in two different systems. One, through the liver, and two, in the muscle and adipose tissue. Now in the liver, what happens is ultimately our PFK1, phosphofructokinase 1, that enzyme that makes fructose 1,6-bisphosphate, that is skipped. So when that's skipped, we have created this dihydroxyacetone phosphate, but then again we have to deal with that glyceraldehyde to make glyceraldehyde 3-phosphate. So by skipping PFK1, because if you remember, PFK1 was our glycolysis enzyme that made fructose 6-phosphate and fructose 1,6-bisphosphate. Well, PFK1 skips that step And so our phosphorylated fructose is phosphorylated at carbon number 1. So fructose 1-phosphate aldolase breaks that into our two molecules. Only one of them is phosphorylated, so we have to employ that other phosphate, or that other kinase. Now, our total oxidation of fructose to pyruvate does give us a net 2 ATP and 2 NADH, because we had to invest 1 ATP there and 1 ATP there. All right, so to contrast fructose metabolism in muscle and adipose tissue with the liver, well, we're using a different enzyme. Now, this is an enzyme that we're a little bit more familiar with. This is our hexokinase. So again, hexokinase means, well, our prefix or our prefix is hexo, meaning 6. So we can use basically any 6-carbon sugar. So we're going to be able to phosphorylate that. One caveat, though, is hexokinase has a low affinity for glucose. We have a high Km for it. So a high Km means a low affinity. Now, what are kind of the consequences of that? Well, this isn't a huge problem unless the fructose concentrations are extremely high, in which case it's going to be metabolizing it at kind of a, maybe it's safest to say, not as fast as it needs to be metabolized. Now, galactose metabolism. From a structural standpoint, galactose and glucose are C4 epimers of one another. I'm going to draw my glucose structure, my ringed glucose, which was down, up, down, then CH2OH. So this is carbon number 6, 5, 4, right there. So glucose, galactose. So we've got galactose, CH2OH. There we go. The difference is just this carbon right here. I'm going to circle it, and right there. So the positioning of that hydroxide group. Now this is a major dietary source of lactose. A major dietary source of galactose is from lactose, because lactose is a disaccharide of glucose plus galactose. Now, there are a number of different reactions that we have to go through in order to get galactose to enter the glycolytic pathway. It's kind of crazy, because you would think that, oh, well, it's just the positioning of one hydroxide group. Shouldn't that just be able to spin around or something like that? Not really the case. So what we have to do with galactose is, first, we have to phosphorylate it with the enzyme galactokinase. That is going to produce galactose 1-phosphate. Now, that galactose 1-phosphate is going to be, I guess, acted upon in conjunction with a molecule known as Udp glucose. So this is a uridylation event. So we're taking this UDP from glucose, and that's uridyl diphosphate, and that UDP is basically taking the place of that phosphate on galactose to make UDP galactose. Now, once you've formed UDP galactose, then what it goes through is, well, it goes and is treated by the enzyme UDP galactose 4 epimerase. So what that is going to do is take UDP galactose and make UDP glucose. Now that UDP glucose has effectively two different fates. One fate is that it goes to be stored as a part of a glycogen molecule. Another fate is that, well, it's acted upon by UDP glucose pyrophosphorylase, that UDP is removed, and you produce glucose 1-phosphate. That glucose 1-phosphate can undergo be treated by phosphoglucomutase to make glucose 6-phosphate. Essentially, what I want you to take away from this is, well, in order to go from galactose to glucose, we have an expense of 1 ATP that will get us all the way to our glucose 1-phosphate. Now, what that means is that if you input galactose, yeah, there are other steps along the way, but our net yield is 2 ATP and 2 NADH. And that might sound like, well, that can't be right. But remember, this step right here, galactokinase, what are we doing? We're taking a 6-carbon sugar and we're phosphorylating it. When you input glucose into glycolysis, you have to invest your first ATP to take a 6-carbon sugar and make a phosphorylated glucose. So we've got a number of steps here where we're going from something that's not phosphorylated to something that is phosphorylated. And yes, it does have to undergo a couple of different steps, and there are metabolites that are involved in this process, but we don't really need to focus on that. Or we don't need to, those are replenished through other systems. Okay, Mannose metabolism. Mannose is the C2 epimer of glucose. So we've got galactose, which is the C4, and mannose, which is the C2 epimer of glucose. Now this is an important part of, or an important component of sugars found in glycoproteins. So those are proteins that have sugars conjugated to them. Now, they're not a major energy source because they're a very minor dietary component. We're going to require 1 ATP molecule. What we're going to do is we're going to take mannose, again, a 6-carbon sugar, and the first thing that we're going to do is we're going to phosphorylate it. We're going to make mannose 6-phosphate. Again, this is analogous to glucose becoming glucose 6-phosphate. Then we're going to use an isomerase. And that isomerase is going to make fructose 6-phosphate. So think about the phosphorylation of glucose. Well, you got to phosphorylate it, then you isomerize it, and then you phosphorylate it again. We're doing almost the exact same thing with mannose. Mannose goes to mannose 6-phosphate, gets isomerized, making a 6-carbon sugar that's been phosphorylated as well. Then we can go on through the rest of the glycolysis process. Our net yield is 2 ATP and 2 NADH. Now, what that shows is that, or to reflect upon that, we've got mannose and galactose that really aren't that radically different from the results of glucose. Now, our major pathways in carbohydrate metabolism, we've already talked about glycolysis, we've talked about gluconeogenesis, we've talked about the citric acid cycle. Now what we need to move on to is glycogenesis and glycogenolysis. So this really captures what's taking place. We're taking glucose and we're building up glycogen, or we're using glycogenolysis to break glycogen down into individual glucose molecules. Now, glycogen is stored in the liver and in the muscle. Muscles use glycogen to provide ATP. Liver uses glycogen to help maintain glucose levels. So what's a healthy blood glucose level? The liver is responsible for managing that. It's going to be your kind of check to see, well, where are we? Now, glycogen metabolism is carefully regulated so that sufficient glucose is available for the body's energy needs. Now, this is primarily regulated by 3 hormones: insulin, glucagon, and epinephrine. Those 3 hormones work in conjunction to help maintain a healthy blood sugar level. Glycogenesis, so glycogen synthesis, occurs after a meal when blood sugar levels are high. Now, this is a 3-step process. Step 1, you take glucose 6-phosphate and it undergoes an isomerization by the enzyme phosphoglucomutase to make glucose 1-phosphate. This is a reversible reaction. All that we're doing is we're taking a phosphate off of one place and putting a phosphate on another place. So if you look in our first step, glucose 6-phosphate, our enzyme is phosphorylated at a serine residue. Our sugar is also phosphorylated. That causes when that's the case, then our phosphate is transferred from our enzyme onto carbon number 1 of glucose. So that leaves our glucose is at one point in this process, it's phosphorylated in 2 positions. Well, the enzyme has to replenish its phosphate, and what it does is it takes the phosphate group off of carbon number 6 to recover our phosphorylated enzyme. Now, step number 2, glucose 1-phosphate is utilized by the enzyme Udp Glucose Phosphorylase. Now, what this consists of is taking a molecule of glucose 1-phosphate and UTP, which is uridine triphosphate, and it's going to remove these 2 phosphate groups to release a PPI molecule, and the subsequent result is UDP glucose. Now, what that results in is our UDP glucose molecule is more reactive than glucose. So this is driven by the energy that is required, the energy that is involved in the formation of this is through the hydrolysis of pyrophosphate. So cutting this off, breaking that bond is what drives this process. Now, glycogenesis is where we have UDP glucose that's added to kind of a glycogen primer. So our first enzyme in this, of the 2 enzymes that are used, is an enzyme known as glycogen synthase. Now, what glycogen synthase is going to do is it's going to see a glycogen molecule which is already present in UDP glucose, and it's going to add glucose onto this chain. What's going to be released from this is our UDP molecule. Now, what this is going to do is it's going to form a straight chain of glycogen, or a straight glycogen chain. So this is going to form alpha 1,4 glycosidic linkages. So if we look at our glycosidic linkage right here, this is carbon number 1, this is carbon number 4, so an alpha 1,4 linkage. This is an alpha linkage because carbon number 1 here has hydroxide group pointed downward. So this is another 1,4 linkage. And it just goes on and on and on. Now, another enzyme in this process is commonly known as the branching enzyme. What this is going to do is it's going to take that long chain on a glycogen molecule, and it's going to create an alpha 1,6 branch by transferring oligo groups from the non-reducing end to an internal location. So that's going to make a 1,6 linkage off of our glycogen molecule. Okay, so now we're going to switch gears a little bit. Now we're talking about glycogenolysis. Glycogenolysis is where we're taking a glycogen molecule and breaking it down. So this is a 2-step process. And what this process consists of is taking a molecule that kind of looks like this, where you have all these potential branch points, and breaking it down into individual glucose molecules. So what's important to recognize is this straight chain right here has a different linkage than what is right here. This is an alpha 1,6, this is an alpha 1,4 linkage. Enzymes are going to treat those differently. So the first step is basically you're going to be able to cleave off glucose molecules, but the enzymes that are involved in cleaving off those glucose molecules are going to sense, more or less, where a 1,6 branch point is. And it's going to stop within 4 glucose residues of that branch point. This is illustrated in the next image. So what this shows us is basically our first reaction. We've got a branched molecule. And this chain right here, the enzyme that is involved in cleaving off individual glucose molecules is going to see this and say, nothing but 1,4 linkages. I'm going to work through those, cleaving all of those off, and I'm never going to really sense anything differently. However, that same enzyme, glycogen phosphorylase, working on these branches is going to be a little bit different. It's going to see this branched molecule and it's going to say, okay, I'll clip off a glucose. I'll clip off a glucose molecule, but then I see that I am 1, 2, 3, 4. I'm the fourth glucose from a 1,6 linkage. And that's where I run into a problem. So I will have to stop right there. So I'll cleave that one off, but I can get kind of close to that linkage on the main chain because of the fact that all I see are 1,4 linkages. So if you're working on a branch, glycogen phosphorylase is going to be able to detect more or less where the 1,6 linkages are, where if you're working on a straight chain, you're not going to have that same sort of sensitivity in some respects. So cleaving off individuals and then dealing with branches. So the first part is, well, break individual glucose molecules off in the form of glucose 1-phosphate, then deal with the branches. Now, an enzyme involved in that process is known as the debranching enzyme. And what the debranching enzyme is going to do is it's going to take over where glycogen phosphorylase says, hey, there's 4 subunits or 4 residues, 4 glucose residues on this branch. The debranching enzyme will say, that's fine, I'm going to take the last 3. So the branch point is basically going to be broken off and there's only 1 glucose molecule there. So that former 1,6 linkage, or that former 1,6 link derived branch, has been shortened tremendously. And so that's when another unique enzyme can come in and hydrolyze that alpha 1,6 linkage. So we've got Amyloalpha16 Glucosidase, which takes the 3 residue branch and moves it to the end of a chain. Then we've got Amyloalpha16 Glucosidase. Sorry, it's not a unique enzyme, it's the same enzyme, but with the more or less a different function. And it's going to hydrolyze that 1,6 linkage. It's going to hydrolyze that away. Okay, and so the result of that is that you're going to be able to break this molecule down, and, well, now we've got a fully liberated glucose molecule that was formerly a branch. That's going to be able to go into glycolysis or enter the bloodstream and and alter the blood sugar level. So we've got hydrolysis, then we encounter a branch, we got to debranch it. Then we're going to hydrolyze that branch, and now we've got a straight chain of glycogen that we can go ahead and process because it's 1,4 linkages. Now the regulation of glycogen metabolism is very tightly controlled by a number of different hormones, which I previously mentioned. Now the degradation of glycogen is regulated by glucagon, released from the pancreas when blood glucose levels drop. So glucagon is going to stimulate breakdown of glycogen. It's going to say we need more, our blood glucose levels are low, let's get some glycogen, let's break down some glycogen so we can get some glucose 1-phosphate that can undergo an isomerization or be treated by a mutase to make glucose 6-phosphate so we can go through glycolysis and get some energy. Epinephrine is initiating a flight or fight response, and that's going to stimulate the degradation of glycogen, whereas insulin, which is synthesized in the pancreas, is going to stimulate the production of glycogen. It's saying there's enough sugar already in the blood, so let's go ahead and make glycogen. Let's store this glucose for later on. Now, epinephrine and glucagon favor the degradation of glycogen through what's known as a cyclic AMP cascade. So here is basically a crude representation of that. And what I'd like to bring your attention to is this right here, glycogen. Okay, now what we have are receptor proteins for insulin, epinephrine, and glucagon. This figure shows, well, are these receptors are they activating or inhibiting an enzyme known as adenylate cyclase? The epinephrine receptor in the muscle is activating adenylate cyclase. The glucagon receptor in the liver is activating adenylate cyclase, and the insulin receptor is inhibiting adenylate cyclase. What adenylate cyclase does is it takes an ATP molecule and it mates cyclic AMP. Now, cyclic AMP is going to activate a protein kinase that will go on to activate phosphorylase kinase. This will also activate, or when phosphorylase kinase is phosphorylated by this upstream protein kinase, what it is able to do is activate glycogen phosphorylase, which glycogen phosphorylase is going to lead to the breakdown of glycogen into glucose 1-phosphate. So there's all these different factors at play. I think that one way that you can kind of look at it is insulin is going to encourage glycogen synthesis, whereas glucagon and epinephrine are going to encourage breakdown. Now, once this cascade is turned on, glycogenolysis leads to the release of glucose in the blood, and that's going to happen very, very rapidly.